to the DNA through the action of the primase enzyme. The thing to also remember is that this is only going to be a couple of primers. This is only be a couple of nucleotides. So we're adding, yes, RNA nucleotides, but we're adding maybe 5 to 10 RNA nucleotides. So I've inserted the numbers 5 to 10 through there. Now, once this has occurred, so after this, primer sequence has been added. DNA polymerase, which of course was the major enzyme, correct? DNA polymerase comes in and takes over the process by adding new DNA nucleotides to the growing strand. Remember that DNA polymerase can only add to the 3' end. And depending on the type of cell, the polymerase is able to add about 500 nucleotides per minute. 500 nucleotides per second if the cell is a bacteria, or about 50 nucleotides per second if it's one of our cells. And you would think, well, if our cells are more advanced, why is it not faster? A couple of reasons. Our stuff is a little bit more thick. We take a little bit more caution with it. Prevent mutations. This is how a mutation would arise. A mistake in this stuff. Good? Okay, so let me show you this picture, which you have already seen before. I had it in the book just a while ago. One thing you'll notice about this, though, is that this throws in the fact that you're using DNA polymerase, right, to connect it together. It's the enzyme that adds to the 3' end. And what do you notice about those nucleotides that are coming in? How many phosphates do they have? 3. How many phosphates does this nucleotide with the A base here have? Just 1. So that means that you're losing how many? Oh, and then they're drawn out right there. There's the loss of 2. Do we see that? Okay. Now, what do we know about something like ATP, if this was an A? High or low energy? High energy. And how about this? Like ATP over here, would that be, or I'm sorry, this is AMP now, because there's only one phosphate attached, right? High or low energy for AMP? Low energy. So that means that you had a lot of energy, and now you don't have a lot of energy in the molecule. That means energy was lost, correct? That energy that was lost was the energy that allowed the polymerase to actually connect stuff together. So what I'm saying, for those of you who are still with me, that the nucleotides that come in their triphosphate form are self-powering. These 3 phosphates that are right there provide the energy to connect them together, to bridge the nucleotides that are neighboring together. How many phosphates do you see in this nucleotide with the T right there now? Just one. There used to be, though, 3, correct? You lose some phosphate, you lose some energy. That means that you're using some energy somewhere along the way. The energy in these phosphates, in this nucleotide triphosphate, has been lost. It's been used to connect it all together. Back in the day when we did glycolysis and cell respiration, y'all remember that stuff? And you made like 38 ATPs, right? If you were here this past weekend at the thing, you saw that that's what we did. We talked about the production of the ATP and cell respiration again. You make a lot of ATP. And we've always associated ATP with being what? Exercise, movement, brain function, right? Now you're saying that that's not just it. You need energy just so you can replicate your cells. The building blocks of your DNA start out in a ATP or TTP or UTP or GTP form. Do you understand what this implies? You don't just need energy to go to the gym or to think about something. You need energy if you're just going to sit there and replicate your cells. Have you ever heard that it takes so many calories a day just to survive? It's like 800 to 1,000 calories a day just to function, just to live, sitting on the couch doing nothing. These are reasons why. It takes energy just to build new cells. All of your DNA are based on ATP and GTP and CTP molecules. Good? Question? Because if you have a nucleotide triphosphate, which is right here, it's high energy. Now it's got one phosphate. It's low energy. If something has a lot of energy and then it has no energy, that means the energy has been used. That's like saying that I had $1,000 in my pocket yesterday, now I have 20 bucks. What do you think happened? I spent it. To do something I didn't want or that I naturally couldn't do. I wanted to go on the Ferris wheel yesterday. And I naturally wouldn't go on a Ferris wheel, so it cost me like $980 to go on a Ferris wheel. It's the same principle. Do you use the energy and those triphosphates to power the coupling, the connection between those two nucleotides that you see right there? This connection is new. It wasn't there before. You need to power it. As those phosphates are lost, it powers it. Breaking ATPs and GTPs, breaking triphosphates down into monophosphates releases a bunch of energy. It's like taking a gallon of gasoline and putting a match to it. You're going to release a lot of energy. Good? Okay. So what we have seen is that DNA polymerase takes over after RNA primase. You start adding new nucleotides, and those nucleotides are self-powering. Now for tomorrow, we're going to tackle this concept again, that DNA polymerase, its issue with only being able to add to the 3' end of the molecule, and what this implies for actually elongating a molecule. Because as you could think, it might become a little complicated since both strands are going in opposite directions. So I want to show you one last picture real quick, and then we can be done if you would like. You have a replication fork here, correct? You actually have 2 replication fork. Here's your origin of replication. And you start going to the left and to the right. Agreed? Now, for your last like 5 minutes, you just kind of have to focus with me on this. Do you see how this strand right here is called the leading strand? Do you see any breaks in it? No. Okay, so right away, that means that this strand has to be, this piece has to be a 5' end, and this has to be a 3' end. Right? Because you can add to the 3' end continuously with no problem. That's why it's called the leading strand. There's no problem with it. If this is 5 and 3, then what could you say about this end of the molecule as opposed to this end? This is 5 to 3, that means that this is the 3' and this is the 5' end. Right? Because the 2 are going to be anti-parallel here. Good? So again, if this was 5 to 3, this would be 5 to 3. Agreed? Yeah. 5 to 3 there. So 5 to 3 there. That would make sense. Dark blue line would be 5 to 3. That means that this guy, right, this is 5 to 3, this would be the 3 to 5 direction. But can you add to the 5' direction? So here's what, and this answers Lindsay's question a little bit. You have to go forward, which you can only add in the 3' direction, correct? This would be 5 to 3 going this way. So the enzyme would then jump ahead and go backwards. See, it started right here at the replication fork, replicated it in the 5 to 3 direction, jumped ahead, 5 to 3, 5 to 3, This is called the lagging strand because you've made a bunch of little pieces.
So is there more than one? Obviously, there's more than one enzyme working.
There's got to be multiple copies of the DNA polymerase that are actually running this reaction. And you see all the little red pieces right there?
Yeah.
Those little red pieces are going to be what types of nucleotides, DNA or RNA? RNA. It's because they were added by primase initially.
Oh, is that like the RNAs?
So the polymerase, yeah, the polymerase can only really start off with RNAs being like the front, several nucleotides. And so there's your polymerase action, the little light blue line. And you can see that how, if it's the leading on this, then it's the lagging there. And if you spend enough time thinking about it, and this is what I want you to do tonight for homework, is I want you to go through those sections in chapter 16 and read over this. You'll see that if this was the leading here, then the other leading would be over here. And it actually makes sense if you think about the 5 degree orientations of everything.
So they're just always opposite.
Mm-hmm. Now, after we get this settled, of how all this polymerase is able to copy and do all of these stuff, and we have our 2 strands that are, if you will, disconnected, then we have to talk about how we're going to glue all the sections together. And then make sure that there's no errors in it. Make sure that the copies came out perfectly. In other words, if this was a T, that this was an A. It has to be perfect. Otherwise, that could lead to mutations, which could lead to a slew of events. Cool? Question?
If the polymerase needs an A, for example, does it just like grab an A?
Because there should be tons of them around.
Okay.
In theory. Now, that would only happen, that would only be in theory, that would only be true if the individual has encountered enough like food and objects, like to provide those ATPs and GTPs. But yes, and usually like if you were going to cut off activities like from a lack of food, you would certainly not cut off activities that would involve like doing this stuff. I mean, this would be like the last thing to go. This is like breathing, like this is vital. You know, if you don't have food, it's like, well, you're just not going to walk today. But like this, you got to have this. It's like basic. It's essential to keep it. Cool. So a little bit more of this tomorrow. That means that on Wednesday, Thursday, and Friday, and maybe even a little bit tomorrow, we're going to start protein synthesis, which is just a tweak of this a little bit in a way. And then probably finish that up on Monday. And I fully expect to have your test over this stuff on Tuesday. Cool. Before you leave for the break.
I am actually flying out on Friday.